Okay, so this next video is about proteins. So we're on our third class of macromolecules. So we did carbohydrates, we did lipids, and now we're doing proteins. I'm going to do a different color here for your proteins and solids concept 5.4. And so proteins are also polymers. They are chains of smaller molecules called amino acids. And proteins, as it says here, have many structures, and that means that they have lots of different functions. And so there's just kind of a big accumulation of various things that proteins can do. So they account for more than 50% of the dry mass of most cells. So they are a huge component of cells. And as it says here, there's a whole list of functions. And so I put this table, it's table 5.1 from your textbook, but it gives you a kind of an overview of the different functions of proteins, and I'm just going to go over them here. So enzymatic proteins, they act as enzymes. Most enzymes are proteins, and what do proteins do? It says that selective acceleration of chemical reactions, so they speed up chemical reactions. So like, for instance, digestive enzymes help to hydrolyze polymers, that means break down by adding water in your food. All right, so they speed that up. Some proteins are structural in nature. They offer support. So some examples of that: silk fibers and spider webs. Collagen and elastin is in our connective tissues. Keratin is found, a very tough, hard protein that's found in horns. It's also found in hair and feathers. Third class, storage proteins. So what do they store? Kind of like, remember, energy storage for carbohydrates. Storage proteins, they store amino acids. So they store chains of amino acids. So ovalbumin is the egg white. Casein and milk store amino acids as protein. Transport proteins, as they're called, transport other substances. An example is hemoglobin. So hemoglobin is a protein in our blood that transports oxygen. So this is proteins that transport molecules across the cell membrane. We're going to see that cell membranes have proteins embedded in them. So these guys, I'm going to put here, transport O2 in the blood. Hormonal proteins, so some of our hormones are proteins, and so hormones in general are released from one part of your body and go to another part of your body. So it's a way for your body to communicate, so they coordinate activities. And so insulin is one of those that help regulate the sugar in your blood. And so we'll talk more specifically about that later. And receptor proteins, these are proteins in your cell membrane usually that says responsive cell to chemical stimuli. What that means is, if this is your cell, you can have a receptor protein in the cell membrane, and you can have another molecule, like maybe a hormone protein, bind to that receptor protein, and what that does is cause a change to occur inside that cell. And so that change would not occur if that molecule didn't bind to that. So nerve cell receptors are examples of that. It can cause a change in the nerve cell by having a stimulus bind to it. Contractile and motor proteins are involved in movement. So in our movement of our muscles, there are two major proteins called actin and myosin that can help our muscles to contract. In cilia and flagella, in single-celled organisms, they are contractile proteins that allow the cilia and flagella for the movement of even single-celled organisms. And lastly, defense proteins protect against disease, and these are antibodies in your immune system. And so they help to combat bacteria and viruses that have infected your body. So that gives you just kind of a general overview of the major types of proteins in terms of their function. So let's turn the page and look at more how enzymes work. So enzymes are a type of protein that act as a catalyst. They speed up chemical reactions. Act as a catalyst. This is anything that speeds up a reaction. So for instance, what examples of catalysts are heat? Heat can speed up reactions. Light can speed up reactions. And also enzymes. So enzymes, I wanted to show you heat, light, enzymes. The enzymes are a type of catalyst. It's a biological catalyst found in living things. And they can perform their functions repeatedly, which means that they can be reused over and over again. So functioning as workhorses that carry out the processes of life. So this picture here gives you a general overview of how an enzyme works. And so we talked with carbohydrates about sucrose, table sugar. And here I want to talk about the breaking down of table sugar. So here we have your two monosaccharides hooked together. So here's your substrate, the thing that the enzyme is going to work on. So you have glucose and you have, I'll do red, you have fructose. That's the other monosaccharide that sucrose is made out of. All right, and they're held together here, and collectively it's a disaccharide called sucrose. So what happens is when you eat sucrose, so I talked about how you cannot use a disaccharide itself as energy. We have to break it down, and so into this respective monosaccharides. So in our digestive system, we have enzymes that work to break down molecules. And enzymes are really, really specific. So notice here it says the enzyme, and notice this enzyme right here has this cutout kind of. If we look at the shape, this is the part of the enzyme that's important in the shape. That shape is complementary to the sucrose. And so notice here they're showing you that sucrose fits right into that shape here that we just looked at here. So you have your sucrose in there. And so this enzyme is specific for breaking down this particular substrate, sucrose. And so that's that. And and it's only used for that. And so notice here, there is one typo I wanted to show you here. It says, notice the substrate here, sucrose, and then it says the enzyme. It also says sucrose. That O should be an A. It should be sucrase. All right. Ase on the end of something means it's an enzyme. Ose means it's a sugar. So here we have, it fits in there into the sucrase. And then remember that enzymes speed up chemical reactions. So how are we going to break this sucrose down? We're going to add water. That's hydrolysis. We're going to add OH and an H to one, and then OH to one and H to another to break that bond. And so now we can see here that these two, we don't see that bond anymore between them. See that bond right here? It's no longer there. And so now what happens by adding that water? It breaks that bond. Let's write that that's hydrolysis. And look at what happens. Hydrolysis, the adding of water. I'm going to make water blue here. The enzyme releases. Now you have two monosaccharides, glucose and fructose. The glucose now has the OH that came from the water, and the fructose has the H here, and they are no longer bonded together. And so now there are two individual monosaccharides. But notice here the enzyme is not used up, so it didn't get used up in the reaction. It still goes back to the same shape, and that enzyme can be reused. That's why it can perform their functions repeatedly and can break down the next sucrose and so on and so forth. And so that enzyme can be reused. And so we have glucose and fructose as monosaccharides, and these then, remember, in our digestive system can enter the bloodstream, be taken to the rest of the body, and these can be used for energy or for building blocks for other molecules. All right, so that's how an enzyme works, and that's a major class over here of proteins and that we're going to talk about a lot during this class is enzymes and their function. And there's a whole special chapter on it, and then even within chapters there are examples and things like that we'll use. So enzymes are a type of protein. Proteins are polypeptides. Poly means many, and peptide is referring to an amino acid, so many amino acids. many amino acids. So polypeptides are polymers of amino acids. A protein consists of one or more polypeptides. So what does that mean? You can have multiple chains of amino acids. So before we build on that, I'm going to come back to that about it consists of one or more polypeptides and later on in the notes here. So let's just look at the basic structure, and that is with the amino acid. So the amino acids are the monomers, so amino acid monomers. They're organic molecules, which remember mean that it contains what? Carbon, right? And so what two functional groups does it have to be an amino acid? See if you can think of it. Pause it if you need to. I'm going to give you the answer here. So this amino acids are organic molecules with, you should have carboxyl, and in any order, and amino groups. So carboxyl, remember, is C double bonded O, OH, and amino group is NH2. Amino acids, there are different types, and how they differ in their properties due to differing side chains, and we call the side chains R groups. And so we have a choice of 20 different amino acids. Cells use 20 amino acids to make thousands of proteins. So just like the letters in the alphabet, A, B, C, D, E, F, G, has 26 letters, right? But how many words can you make? I mean, there are thousands of words. How do the words vary? They vary in the number of letters as well as the order of the letters. And so therefore, with the same 20 amino acids, you can make thousands of different proteins because you can have the length different and the order in which the 20 amino acids are hooked together can be different. And so if we look at the basic structure of an amino acid, there's an asymmetric carbon atom right here in the center. So this one's asymmetric. It has the 4 groups it's attached to. And so it's always going to have the carboxyl group. So right here, this is your carboxyl group. Over here, it has the amino group. It's always going to have a hydrogen. So that's 1, 2, 3 things that are true about it. And then off the 4th, you're always going to have an R group. And the amino group and the carboxyl group are always on the ends, here, left and right sides. And so you're going to have an R group and the hydrogen. And the R group, that's what differs. So there's no such atom. There's not an element R. So that just stands for radical, actually. But it just means that there's some kind of group of atoms. And so that is in that R group to make one amino acid different than the next amino acid. So let me show you some examples. So for these, notice that these are all different types of amino acids. You do not have to memorize these. And so this one's name is glycine, and they always abbreviate it usually by the first 3 letters in the name. So GLY is glycine. And notice here it has your amino group, the H, the carboxyl group, and outlined here in white, that is your R group. And I do want to point out something about the amino and the carboxyl group in here. So notice here it says amino group and it says NH3. So what happens is if you have NH2, that's the amino group. But remember, it acts as a base and can accept, whoops, my light shut off. It acts as a base by accepting H+ ions. So what happens is, if this has already acted as a base, it can pick up an H+ ion. So if this has already acted as a base, you can see it written as NH3+. So this has already acted as a base. And then over here, notice that this is the carboxyl group. If we look at that over here, it's the C, whoops, double bonded to an O. O negative, that's what it says here. Remember that the carboxyl group usually looks like this, but remember the carboxyl group causes the molecule to act as an acid, which means it gives up H plus ions. So if it gives up the H plus ion, it becomes, and it looks like this. So this is still considered the carboxyl group. It's already acted as an acid, and this is acted as a base already. All right, so I just wanted to point that out as far as why it looks different. So you can see here that then alanine is a different amino acid. If I cover this up here, the R group, it looks the exact same as glycine. Valine, if I cover up the R group, looks exactly the same as alanine and glycine. So these right here are your R groups. These guys are called non-polar amino acids. Why? They do have polarity because they have the functional groups, the carboxyl group and the amino group, but they're classified as non-polar because they're classifying it based upon the R groups. All their R groups are basically non-polar. The majority of it is carbon and hydrogen. All right, so when we look at, so that's the non-polar R groups. Then we have down here polar R groups. That means their R groups are polar. So therefore you can see that the R groups themselves have some functional groups, hydroxyl, amino, sulfhydryl, and so on. So those are your polar ones. And lastly, we have electrically charged. It's our third group of amino acids to give us our total of 20. And that means that they either act as a base or a acid by either giving up H pluses or accepting H pluses, and therefore have charges. So these carboxyl groups here in the R group have given up an H plus already, so they have a negative charge. These R groups have the amino group, and they have accepted H pluses already and have acted as a base and therefore have charges, and so those are called electrically charged. So collectively we have 20 different amino acids, and they differ in the R group. So then we have to hook them together to make proteins, and so to make a polypeptide or many amino acid chain. And so amino acids are linked by, and it says blank bonds. They have a special name for these bonds. They are called peptide bonds. And a polypeptide is a polymer of amino acids. So it's one amino acid hooked to another. I'm just going to put AA for an amino acid hooked to another amino acid, etc. So, and they can vary in length from just a few monomers, like this one here, to more than 1,000. I'm not going to sit here and draw 1,000, but you get the idea. So each polypeptide has a unique linear sequence of amino acids. So what makes them different is their length, as well as which of these 20 amino acids and in what order are they hooked together, kind of like letters in a word to make up different words. So the amino acid sequence of polypeptides were first determined by chemical methods. We're figuring out what amino acid was hooked to what amino acids. And so now we actually use, as they're automated, we use computers. There's high-tech computer systems to be able to figure out which amino acid is hooked to which amino acid to make up a polypeptide chain. So let's look at protein. It says protein conformation. You need to know that this word means shape. So whenever you see the word conformation, it's talking about the shape of something. And we're going to see that the shape determines the function. So a functional protein consists of one or more polypeptides twisted, foiled, and coiled into a unique shape. And the sequence of these amino acids, the order of the amino acids in the protein, the sequence of amino acids determines the protein's three-dimensional conformation. So what kind of shape is it going to have? A protein's conformation then determines its function. And so we can look at, it says ribbon models and space-filling models can depict a protein's conformation. Let's turn this over here. And this didn't copy well on your picture, so I'm going to show you this. So this is a ribbon model. So what this is showing you is the ribbon in purple is the chain of amino acids. So the chain of amino acids aren't just linear, but they curve and bend and so on to give the protein a three-dimensional shape. That's one way to kind of show shape for a protein, and this is called a space-filling model, where it shows you the balls are supposed to be the different types of atoms and how they're bonded together, and you can see overall that it has, like, there's a little groove here. So if this is an enzyme, this could maybe be the spot where the substrate binds to, and so it has a specific shape. And so you'll see in your textbook one of these two ways to show shape for the protein. So let's look at how proteins are put together. There's 4 levels of protein structure. So the primary means first. So the primary structure of a protein is its unique sequence of amino acids. So what amino acid is hooked to what amino acid, and so on and so forth. The secondary goes right on here, structure, which is found in most proteins, consists of coils and folds. So it coils and folds and is not linear. Then what comes after secondary is tertiary. The structure is determined by interactions among various side chains, the R groups, and we'll talk about this in just a second. I'm just giving you an overview of that. And the fourth level, kind of layer of structure is called the quaternary structure, when a protein consists of multiple polypeptide chains. So this here I'm going to go over in just a second here. Let's talk about each one of these first before we look at the big picture. So let's take primary structure. The sequence of amino acids in a protein is like the order of, I use the analogy of letters in a long word. So what letters are hooked together determines the word. And so what amino acids are hooked together determines the protein or the polypeptide. So primary structure is determined by inherited genetic information. So it's through your genes. Your DNA determines what amino acid is going to be hooked to what amino acid, and therefore what protein is going to be made. So this is a linear chain of amino acids. Each one of these little balls is one amino acid. So we have many amino acids hooked together. And so basically your primary structure is an amino acid hooked to an amino acid hooked to an amino acid, and so on and so forth. So if I were to do it kind of like a ribbon structure with just my pen here, it would just be linear. So this is like a long chain of amino acids, one long chain, this string here, let's say. All right, so that's the most basic way that you can think of a protein. So that's your primary structure. Then, so that's the first one. The second one is the secondary structure. The coils and folds of secondary structure result from hydrogen bonds. So here we go into hydrogen bonds, between repeating constituents of a polypeptide backbone. So I'll talk about this here. Typical secondary structures are a coil called the alpha helix and a folded structure called the beta pleated sheet. And so when we look here, what we have is a chain of amino acids. So I went here just amino acid hooked to amino acid hooked to amino acid. Actually, to hook them together, if we look here, I'm just going to show you that up here. If we draw just one amino acid, all right, it has the 4 bonds, right? So it has an R group, an amino group, and a carboxyl group in the hydrogen. So that's what one amino acid looks like. So if we're going to bond another amino acid to this, so what happens is I'm going to draw a second amino acid. So I have the carbon, then I have my NH2, and then I have my R group, and then I have my carboxyl group, and then my hydrogen. So this is amino acid 1, and this is amino acid 2. So in order to hook them together, what process do I need to hook them together? Dehydration, right? Losing an OH, so we lose the OH here and the H here. Molecule of water is lost, and guess what? Now this carbon is bonded to this nitrogen, and that is your peptide bond. And so then you could hook another amino acid by the same way, and so on and so forth. I wanted to go through that with you to show you that when we're looking at a chain, this is a chain of amino acids. So what you have to do is look at here. If we look right here, here's the carbon, here's its R group, here's the hydrogen. This is the, was once part of the carboxyl group, and over here this was the amino group. And so this bond right here is this amino acid right here bonding to the beginning of the next amino acid, and this right here is the beginning of the next amino acid right here. So if we look here, asymmetric carbon atom, R, H, amino, carboxyl. And so therefore this is the to the next amino acid right here, and so on and so forth. So this is truly a chain of amino acids. That's what I wanted to show you here. So what it's showing you, it's kind of hard to see here, this is supposed to curve around here, so pretend like there's amino acids here. So that's what you're looking at, is these are called the beta pleated sheets. So it's like an accordion here, and it can wrap around and the beta pleated sheets, and then you have right here the dots are your hydrogen bonds. All right, from the positive hydrogen of one to the oxygen of another. And those can hold those together, all right? And so therefore, instead of having just linear, they can actually hydrogen bond with one another. Also, depending upon the amino acids that are part of the chain, they can hydrogen bond in a helical shape. So again, they outlined here, this is one amino acid bonding to another amino acid, so you get the chain of amino acids, but in this case they're hydrogen bonding this way, and that's causing a helical shape. So so if I talk about it in terms of just this ribbon here, what can happen is we can redraw it just as a straight line to kind of see the difference between this and this. So down here, like this one here, they're kind of showing you like this, with the hydrogen bonds between them like this. Or what this is showing you, what I was trying to show you here, is that they form these pleats, and then it can curve around and form pleats like this, and the hydrogen bonds are between there like this. Whoops, I'm sorry, I'm off the picture there. So it can curve around, so it's actually connected together to show you the big picture there. Or it can be helical. And so, as well, so then the hydrogen bonds are connecting this way. All right, and so that is your secondary. So you have, so if I were to draw this, you could have the straight line, which is the chain of amino acids. You could have part of it do an alpha helix like this, and then you can have beta pleated sheets, and it can come around here, beta pleated sheets, and the hydrogen bonds between there. All right, and so that is your secondary structure. So then your third is your tertiary structure. So tertiary means third, and this is determined by interactions between your R groups rather than between the backbone constituents. The backbone means that's the actual part of the line in between, like the C's and the N's here, not what's hanging off the R groups and the H's there. So it's actually interactions between the R groups. And there are different types of interactions: hydrogen bonds, ionic bonds, hydrophobic interactions. So strong covalent bonds called disulfide bridges may reinforce the protein's conformation. So these guys are very strong and make proteins that have a lot of these disulfide bridges are very, very strong in nature. So let's turn it over and look at this. So this is supposed to be your ribbon model, your three-dimensional look at the tertiary structure. So it involves the primary, which is the chain of amino acids. Secondary was the coils and the pleats, and the tertiary is the bending, where they bend around. So, for instance, every single time there's a curve in the shape, it's due to interactions of R groups. So when we look at this, you have right here, when it bends, like right here, if I were to enlarge that, it bends because the R groups of the amino acid right here and the amino acid right here interact in some way and are attracted to one another, which helps stabilize this curve. And so let's look at some examples of this. So first of all, with this ribbon here, you have to understand that this ribbon is a chain of amino acids. So right here in this spot where this CH2 with the OH is coming off, there's an amino acid in that spot. So I'm going to write AA here. And then the rest of this is chains of amino acids. So there's an amino acid here. And then right here in this spot, there's an amino acid with this R group coming off. Then you have the chain of amino acids, chain of amino acids, chain of amino acids. And then you have a specific amino acid in that spot that has this R group coming off. Then you get the point here that there's a chain of amino acids, and then there's an amino acid here that has this R group, amino acid here that has this R group, chain, amino acid here that has that R group, and keep on going with the chain, and you have an amino acid here. And so therefore, what I want to point out is this amino acid has this R group, and way down the line, it could be hundreds of amino acids later, this amino acid has this R group. And guess what? These two R groups interact and make a hydrogen bond. So let's write here that this is the formation of a hydrogen bond. Right here, that's the dots there. And so what we see here is this is one R group, and this is second R group of the two different amino acids way far along. And so therefore what that does is help, because these R groups interact, help stabilize and cause this curve to happen. So it's no longer linear because of the attraction between the H's and the O's there. Notice for this one, this amino acid, and this is its R group right here, and this amino acid, and this is its R group. Notice that they're both hydrophobic. They are both made out of just C's and H's, which means they want nothing to do with water. This is called, these two R groups interacting, this is the second kind of interaction, are called hydrophobic interactions. All right, so these guys, what's happening is the R groups are, nonpolar. I almost wrote polar, getting ahead of myself here, are nonpolar, which means that they arrange themselves away from water. So remember that proteins and living things are in water-based substances. So you have a water-based solution around here. And so guess what? Water is attracted more to itself and other polar R groups and so on than the hydrophobic ones. So what happens is the hydrophobic ones kind of fold inside. They separate out kind of that, just like that oil and water example that they separate out. The hydrophobic ones, the R groups orient themselves in the interior interior, the middle of the protein, away from the water, because the water is more attracted to the other parts of the amino acids than they are to the hydrophobic R groups here. And so those are called hydrophobic interactions. So these guys aren't bonding to one another. They're just arranging themselves in the interior of the molecule and away from water. And I'll write here in the interior of the protein. I'll add to that, into the interior of the protein. Okay, so then let's look down here. We have here, let's look at these two here. So this is the R group of this one, and this is the R group of this one. These are the electrically charged. This is a nice positive charge. This is a nice negative charge. So these guys are attracted and can make an ionic bond with one another. And so therefore they're attracted based upon the opposite charges. And so these guys, this amino acid and this amino acid, separated by, could be 30 different amino acids here, but the R groups interact and stabilize this curve. So this curve is stabilized by these attractions as well as the hydrophobic interactions. And then we have another curve because these guys, the R groups, are electrically charged and are attracted to each other. And then lastly, we have these two. Oops, I forgot to put an amino acid here. This R group and this R group. Notice here, these S's are bonded together. And so this is called, it's right over here. This is the fourth. It's called a disulfide bridge. The disulfide bridge is why it's strong. It has a covalent bond between two sulfur atoms of the sulfhydryl group, of two sulfhydryl groups. Remember which was SH. These guys, let's write, are it's a very strong bond. It's a covalent bond between them. The amino acid that has that occurs between two cysteines. Those are the amino acids. And an example is these guys are found in hooves of animals, nails, horns. So you can see very, very strong. I have a lot of those. So it makes them very, very tough protein. And so that's your tertiary structure, is the curves and the bends of that. And so here, how I would draw that with my little drawing here is, remember you had your chain of amino acids, you have your beta pleated sheets, let's say, and then you have your alpha helix here and the beta pleated sheets here. And so these guys are hydrogen bonded together and these guys are hydrogen bonded together. So this would be stabilized by R groups here. This curve, an R group and an R group could be stabilized, this curve. And so overall you get this nice shape, all right, because of the order of amino acids, the coils and pleats, as well as the interactions of the R groups. All right, and then lastly, the quaternary structure. This is the fourth. Quaternary means fourth. And this results when, go back to my color here, when two or more polypeptide chains form one macromolecule. So in this case here, an example I'm going to give you is collagen. Is a fibrous protein. That means it's kind of like a fiber, it's string-like. And so consisting of 3 polypeptides coiled like a rope. So if you look here, this is an example down here of collagen. And so, so collagen has 1, 2, 3 polypeptide chains. So each one of these ribbons represents a polypeptide chain. And so this protein, this is one protein made from 3 polypeptides. So one protein made from 3 polypeptides. This is an example of hemoglobin. Is a globular protein, meaning it's not fibrous like this. This looks like thread or yarn, consisting of 4 polypeptides. So this is like one glob where it's 4 polypeptides. It has 2 alpha and 2 beta chains. We don't need to go know that much in depth, but basically right here is one chain right here. This one is hard to see. This one is not connected to this one. It's a second chain, and there are 2 dark chains, 3 and 4. So a way to think of that is 1, 2, 3, 4. So this is one protein made from 4 polypeptides. All right, and so, but it's one protein, and so hemoglobin carries oxygen, and so some proteins are done with in building it and at the tertiary structure. So some are just one polypeptide chain. So this could be a completed protein. Let's say it's an enzyme, but some proteins need to go through this 3 or 4 times to make 3 or 4 different polypeptide chains, and then all 3 or all 4 of them have to come together to make your finished protein. And so collagen and hemoglobin, let's write that this is hemoglobin, are examples of that. All right, and so that is the 4th level of structure for proteins. And this is where I'm going to stop off. There's a little bit left with proteins. It's more application here. I am going to wait until the last video to do that. All right, because this one's already at 41 minutes.